A very good question, and Mark and I might share that. Go ahead, Mark.
Probably the primary one is one of scale. MABs typically have high doses and very, very large material requirements, and therefore will have the battleship-style plant for many of the applications. With vaccines, the doses can tend to be more variable, and the vaccine type of manufacturing is a much smaller scale, probably for the most part on the scale that you could handle with skid-mounted equipment, and you could move it in and out on a matrix basis. Some vaccine processes also have a small number of logical operating units, and therefore would not consume large amounts of a manufacturing facility.
And maybe the other half to the answer is a manufacturer that does both traditional egg-based as well as deep tank. There's still going to be a great deal of vaccines developed in the traditional egg-based technology. Now, this can get very advanced. If you look at heavily automated plants, one of the recent in fact, 2 of this year's Facility of the Year awards were in the vaccine space, one in traditional egg-based, one in deep tank. What you look at there is a much stronger focus on reliability of operation, and of course contamination as well as segregation and isolation is a much higher factor. If you look at H7N9, which is beginning to get some press as a possible pandemic coming out of China, that currently as a wild strain has to be handled at BLS 3+. Which is BLS4 is as bad as it gets. Typical influenza is BLS2 or BLS2+, so that enters into the equation as well. And some manufacturers will simply not choose to work in that space. The other thing that enters into the vaccine business is the whole question of who wants to be in which space. It's very difficult for the companies that focus on pediatric vaccines or seasonal influenza vaccines to also say they are going to make biodefense vaccines. You'd almost have to separate the plants. It has nothing to do with regulatory affairs. It's more of a marketing, commercial positioning of would you want someone making a vaccine that services, say, anthrax in the same plant, regardless of the regulatory issues that makes influenza that goes into kids. Just a question. Some companies deal with it and others don't.
Also, vaccine manufacturing sometimes entails the use of BSL-3 facilities, and that's a case where you would have a matrix-like configuration that would have a segregated arm which would be capable of implementing other unidirectional methodologies that you could enforce BSL-3 when it's appropriate for that particular product.
And just sort of to close this particular question out, we are seeing that the right answer for a number of our clients is really a solution that is hybrid, that in order to make the appropriate optimization for their particular product lines and for their particular business needs, that what they end up with is a facility that really borders on implementing both the traditional stirred tank stainless steel approach for some applications coupled with the implementation of single-use systems. And the configuration of the facility then sort of takes on a little bit of a hybrid type of process architecture as well. So again, as we mentioned earlier, the solutions will vary, and we're beginning to see that quite a bit in the industry. So.
Good question. Yes.
In terms of the production platform, I would like to know your opinion on the use of the plants as platform, considering that the FDA approved the first plant-derived compounds for use in humans a couple of years ago. Do you think that this platform may become important in the future in industry?
Well, I don't know if there's anybody from Medicago sitting in the audience, but they would probably definitely say, oh, yes. I think that the application of the technologies is going to continue to grow. I know that Medicago that I mentioned, they have a facility that is very close to our office, and it's one where there's a tremendous amount of interest in the platform itself. There have been companies that have gone down that path and they have failed. But again, I think that what we're beginning to see from the standpoint of the science is that the viability is occurring and the interest is definitely there.
It sort of approaches the whole subject of cell lines. I think there probably will be additional plant cell lines developed in the future. They have some significant advantages with respect to contamination and stuff like that. The first large jump in productivity in mammalian and cell culture systems occurred from media optimization. They're now beginning to splice out unproductive metabolisms out of cells, and I think you're probably 10 years away from having more specialized cell lines that are created and tailored to the manufacturing recombinant proteins, and so you could see a jump to 20 grams per liter. Whether that occurs with modified plant cells is yet to be seen. There's tremendous amount of potential upside. One of the reasons why I think you'll see a major shift in the vast majority of products to smaller systems is that tremendous increase in productivity where you get 20 plus grams per liter. And there is potential for significant enhancements in purification technology. There was a session this morning on affinity chromatography, and there's active research in that area. And it has a ways to go, but there's a number of routes that could significantly decrease the size of the manufacturing unit operations.
What you have to consider, and this is where the industry often makes the mistakes, is all that matters is getting the product to market at a reasonable cost. A lot of the focus on expressions in sheep or mammals or expressions in plants were initially generated back when you were getting 1/10 of a gram per liter out of mammalian cell culture technology. Now that you can get 5 grams, 6 grams, 10 grams per liter, if you look at a typical cost of goods manufactured, COGM, you hear that expression all the time, or cost of goods sold, COGS, both numbers are a useful number. Small molecule, the number is around 35%, 33% for an old legacy, relatively inexpensive final dose small molecule product. Maybe you have some in the 40%. Bio, the rule of thumb is 14%, but quite frankly, that's overstated. Most bios target less than 10%. There are legacy COGMs out there that are less than 1%. So when the cost of goods manufactured is it 1%, is it 7%, is it 10%, you look at what's the most reliable way of getting the product to market. Only if you need thousands of tons of material, and that's a rare, that's not a therapeutic, would you walk away from a platform in which you know you can make the product. Now, there are certain proteins, protein fragments. It's the same reason why you use microbial or mammalian. It's what can make the product better. There may be proteins that can be best expressed in a totally different platform. One of the messages we always give our R&D colleagues is there are other hosts besides mammalian cells. There are other hosts besides microbial. They are higher yielding at the R&D stage. But when I have a COGM less than 10%, and I've got $800 million in stainless based on a fixed platform, give me the product at 1/3 less yield in a platform we have as opposed to go find the next platform just because the yields are higher, because quite frankly, at those costs to produce it, it's immaterial.
Any other questions?
One of the other questions that came in from the outside, and maybe we address some of those, the trade-off, and it came out both in Jeff and Mark's discussions on the flexible plant of the future is versus fixed stainless. There's an awful lot of process contact, process change, operator intervention. And if you work in a plant in which you're only producing 2 or 3 products, the last thing you need is change. There's an old joke within Medi that if the plant manager always parks in the 3rd spot from the left by the front door, I want him always to park in the 3rd spot from the left from the front door, because anything that changes in that plant still has the potential of impacting the process. These processes are robust in many ways compared to 15 years ago, but you'll find deviations that trace back to a change that you would never imagine could have caused a process upset, and it might take you 30 lots to figure it out. So perhaps we can comment on the balancing act of what judgment factors do we use in looking at, I'm willing to invest more in fixed stainless because it doesn't have a lot of change, a lot of operator interface. When do I make the trade-off of these plants are extremely flexible, but they require a lot of operator interface?
I think there's going to be a considerable emphasis on speed in the future, and I think the single-use systems, plastic systems, provide an ability to move much, much faster. You can move to a scale which is fairly similar to what you will have to manufacture commercially. You would develop at that scale and then go through the clinical trials, moving the process in and out of the facility as required to meet the material requirements for preclinical, early clinical, late clinical, commercial, and launch. The stainless steel systems, fixed stainless steel systems such as the battleships, are very, very difficult and very expensive to build and are not very flexible. The more flexible you make them, the cost becomes exponentially more difficult in terms of adding all of the different supporting piping to make automatic systems. I think you're going to see the industry forced to go to single-use systems for development through commercialization to launch. Now, if the product develops large material requirements and you feel that life cycle is stable and the product will have long durability, then you would use a stainless steel asset, either an existing one. The time to build large plants is probably too large to meet the life cycle of new products. New products will have much shorter life cycles. They will be basically disintermediated by a better product. That's one of the real threats of biosimilars is really biobetters, and the fact that with enhanced analytical capabilities you will identify a product which has a better CQA profile than the existing drug, and then therefore you will have a superior therapy. It's very complicated, but I think single use is really a very important tool for speeding things up and moving things faster and faster through the pipeline.
You can actually put some dollars and some numbers on that if you don't work with these plants directly, because we have to model these production spaces. And the models are interesting. They wind up in a multi-dimensional map that only a computer can visualize. But if you simplify it to 2 or 3 dimensions, let me just carve out a couple of planes in this three-dimensional map of cycle time versus turnover time versus the number of lots that you run. If you take a battleship, as Mark talked about it, if you have a cycle time of 5 or 6 days and you only run one product turnover a year, and your turnover time is also on the order of 5 or 6 days, that plant has a capacity of, call it 75 lots a year, plus or minus. If I have a cycle time turnover time of, cycle time of 12 days, just the cycle time, not the turnover time stays the same, I automatically reduce the capacity of the plant by 50%. If my turnover time is 14 days instead of 15 days and I run 6 products a year in that plant, I can produce 17 lots a year. It's the same stainless. It's the same $550 million price tag. It's the same $140 million a year run rate cost. But in one case, I get 75 lots out of it, and in the other case, I get 17 lots out of it. That's why what Mark and Jeff are describing, that's why you have this huge balancing act. Now, if you look at human intervention versus risk of loss, that's more math. If the plant, ideally a plant that does development work, and typically the larger plants will do phase 3 product because you'd like to do phase 3 product at the same scale as commercial, speed to launch. You don't want to be having to have a debate with the FDA of whether I can scale this from X hundred liters to Y thousand liters. You want to run it at Y thousand liters and eliminate the question. So you run phase 3 in these plants. But as we look at that, and you look at the introduction of the number of turnovers, you would have to leave that type of plant. 10 years ago, we'd leave that plant at 65% utilization. We'd do that intentionally so that we would have the flexibility of being able to move in development products quickly on short notice. Today, you can't get away with that kind of cost. We target 80%. Our CFO would like to target 85%. I've heard folks foolishly target 90%, which is chemical industry standards. It's not happening in bulk bio. But at 80 or 85%, now the plant is essentially fully utilized. Long-winded conversation, but it gets us back to reliability. If I have a plant at 65% capacity and I lose a lot, the cost of that loss is only the incremental material and the labor for the plant. Pick a number out of the air. Is that $2 million? Is it $5 million? It's somewhere in that range. That depends on the operator and what your CFO is willing to allow you to divulge, but it's in that range. If I'm at 85% capacity and that plant's now full, that lot I lost is an irreplaceable lot. One of those lots, typical bio, they're all the same. One of those lots, that bioreactor you're looking at, that has nobody standing around it, it's all run by automation. That bioreactor has $100 million worth of product in it. You don't lose one of those without having calls to the CEO when they're irreplaceable. So that's the disadvantage of pushing the utilization of these plants up into the 80-85% range. And if it's not a commercial product, then I'm short one lot for my clinical trials, which is even worse, quite frankly, than losing a commercial product, because I've only lost one lot of commercial, but I may screw up a clinical trial, which may move a product from first asset in class to second or third asset in class, and its market value is a quarter of what it would have been. So these are very complex discussions if you run operations and development inside a biofirm.
Running manufacturing is all about utilization. There's been a kind of an unfortunate tendency to view cost of goods and cost cutting and all that stuff. But that's all pretty easy and fairly trivial. It's really about what manufacturing assets do you put into place to give you a high utilization. One of the reasons they run manufacturing facilities underutilized is they have to have an ability to handle an upside demand should it occur. Failure to supply a market is a cardinal sin, and you never know when you're going to get increases in demand. We used to wrestle that. If you have a long-term, irreplaceable manufacturing asset like a battleship, you just have to make them way too big and then run them underutilized intentionally. The modern technologies or modern manufacturing capabilities will have to be something that can be expanded very dramatically, fairly readily. And that can be done with the smaller single-use systems by what is termed a scale-out. If on one of the slides you showed all the different phases that a process goes through. You launch a product, and there's usually a blip to fill up the pipeline, and then you recede to a more modest manufacturing capacity, and you wait and see what happens. And in some cases, your market gets kind of slow because somebody finds out that some other cheaper product's just as good, or you find a runaway demand because there's additional indications. And so you have to be able to have a manufacturing capability that you can actually physically expand or contract and still maintain reasonable utilization rates of your manufacturing assets. If it shrinks and your facility can readily handle other products and other phases for developing new products, then that utilization can remain high by using it for other purposes. However, if you get a runaway demand, you begin to scale out your process by duplicating it, which is readily achievable with single use, and building additional manufacturing capabilities. One of the things that's very important in the future is to be able to clone a manufacturing facility to send it overseas. The BRIC countries like to have the manufacturing done internally. And so that is another advantage of the more advanced facility designs, is that they can be literally cloned and shipped overseas.
Yeah, and one point I'd like to make, years ago when we would sit down and start to look at a new concept for a facility, very often in the conversation we would always talk about this idea of expansion capability in order to meet market demands. But it was pretty rare that you ever had the conversation about contraction. And yet with a lot of the companies that we do business with today, that's a very key part of the equation. Especially with CMOs, as they wrestle with how to look at their business model in this new decade. The concept of contraction comes into the discussions from the very beginning, and it's really something that with the new technologies you can readily talk about, you can design for it, and it becomes an integral part of the business philosophy, because inevitably, for a lot of our clients, it will happen.
Yeah. And one of the things you do too, and that's where a lot of the partnering has come into play, for certain sizes, if you need thousands of kilos of protein, if that's the size of the marketplace, you're not going to do that in a small facility with a great deal of human interaction. The risks of loss or unpredictability are too high. But not every product, those are the rarer products in this day and age, especially for new pipelines. You bet some of that, you offset some of that bet by partnering, and every single major bio has developed some form of partnering structure in the last couple of years. Better to be betting these large dollar investments across a risk-adjusted pipeline of 2 companies than 1 or 3 companies. Beyond 3, then it becomes very complex plannings. If all 3 hit at once, you can hardly respond to it. So that's one way of offsetting these bets, but what enters into the equation as well is the sheer scale of what might be needed for protein out of such a plant. Again, the right answer for many modern products may well be small, fixed, or single use. But when someone says, I need X thousand a year of kilos, then you almost are looking at, if I'm going to have to run 50 lots a year out of a 15,000 liter bioreactor, and I have to do that for 7 to 10 years, I'm going to have fixed asset and stainless to do that just because of reliability.
Ken.
Ken Jacobson, CRB. A different way of viewing your $100 million lot, Andy, is that it's only a $100 million lot if it's a sold-out plant, which is what you described, the 80 to 85%, and you have no other assets elsewhere to manufacture. And to put yourself in that position with a 30 or 40% gross margin is one thing, but in a 1 to 14% cost of goods, it's bordering on irresponsible, because if you're not in that position, which is, I've designed plants where we knew going in it was a sold-out plant, and you take quite a different approach. But if you're not in that position, which is the normal case, then it's the cost of goods that's at risk, and you can make up that lot. So it's just...
What's happening, and I've lived through all of this, what's happening is the mentality and the leverages of the small molecule side of the business are overlapping with the large molecule business. And the leverages are dramatically different, but they're being blended together because we have monolithic ways of running a company. Mm-hmm. So while we would all know that I would never wish to, the other thing that's different, not only would you be financially foolish or feel financially foolish if you allowed to run a plant flat out, some of it's timing, because you don't know how big to build to begin with. So in the early stages of the market, you may well be sold out because the market gets ahead of you. But the second thing that enters into the equation is a universal demand for heavy utilization and a totally different balance on sole asset and class versus multiple asset and class. For the audience that doesn't deal with that all the time, what do we mean? There's 5 companies that make a version of Lipitor, and there's probably 12 generics on the marketplace. So I don't mean to pick on any one company or a name, but just Lipitor number 2, brand X, brand Y, pick it, winds up short to the market. Physician can rewrite a replacement script in a heartbeat, both with second-generation products that's virtually the same or a generic that's 5 or 7% less efficacious. So there the industry is taking knowing risks on shortage to supply to max out the utilization of the chemical stainless in the business. The industry is taking knowing risks to reduce costs, either by outsourcing or market penetration. Everyone is. It's not company A or company B. Bio, almost all of us work on products that are sole asset in class. There is no other asset. You don't make Enbrel. Somebody winds up, they can't walk to school anymore, they're back in bed because their joints hurt so badly they can't get out of bed. We make Synergist, pre-meat prevention for RSV. It's the only market on the product. If there's ever a season where I don't supply Synergist, I can do the math. Of the preemies born 32 weeks early, 40% will get RSV. Of the 40% that get RSV, somewhere between 7 and 11% will die. Another 7% will have such severe respiratory infections that for the rest of their life they'll be carrying oxygen bottles around. They'll never play a sport in a school. And another 7 or 10% will have severely compromised but survivable. So you can just do the math. Forget the dollar value of what a season's of that product is worth. I'd wake up in the morning doing the math of how many kids have I just killed. I don't ever want to do that math. That's totally different from a lot of where Big Pharma is. But all those, at the CFO level, CFOs don't deal with these degrees of subtlety. It's a very different place the industry is in today. And that's not that they're wrong. They have businesses whose model is collapsing. All of them do. So it sounds foolish, but in reality, those are the equations you deal with.
Yeah, when we're looking at biosimilars and biobetters, I think that cost of goods fraction is likely to change dramatically for biotech products. You know, so that when you do have, when you aren't the only RSV product out there,
yeah,
it's a different situation.
Different,
at least the whole world's not relying on you. Mm-hmm. But that's small comfort in the lost profit.
Yeah, it's still a big commercial hit,
yeah.
but Janet Woodcock isn't the first one calling you in the morning. Yeah.
Right.
Wall Street Journal might be.
I had another question for Mark on the presentation regarding closed processing and the impact on what future facilities look like, because we're seeing a trend toward people relying on the closed processing to say, well, my process doesn't have to be in a clean room because the environment doesn't impact product quality. It's a non-impact. So the HVAC is a non-impact system now. Potentially doesn't have to be validated. And yet I saw that you still had clean, dirty flows, and I was wondering if you wanted to comment on that, because I was hoping the trend, I was viewing the trend was really trying to go away from that and say if the system is closed, whether it's single use or stainless steel, by the way, no reason a stainless steel system isn't easier to make closed than a single use system.
Well, I'll give you a short answer since they're waving at us to get off the stage.
Our monitor is picking on us.
Sometimes people pick up an enabler and they look at an enabler and they don't really understand what they really can use with it. And single use has been picked up and said, closed system, now I don't have to use a clean room and I can commingle everything together. And that's not the strength of single use systems. If you put it and not put it in a clean room, then you increase the burden of the procedural controls to establish or prove that the system was closed and that primary barrier was in place throughout the life cycle of manufacturing that product. And that's a pain in the neck, okay? It introduces more garbage to the manufacturing. If you use the single use in a contained space, then you don't have that procedural burden to overcome. You have to answer the question 6 months after you made the product if something squirrely is happening in the marketplace. Was the system closed? And that adds more stuff that has to be done during manufacturing. If you have tight physical segregation within spaces, then that overhead is reduced considerably. And it gives you a variety of options to deal with if there is a problem with the system and it is not closed.
The other thing, and Mark put his finger right on it, if you're dealing with something that comes up, the biggest thing you want to be able to do is narrow it down to a lot, a bioreactor or a closed. If it's an environmental issue, then you have to do a search and destroy of, was it one lot, was it 1 week, was it 1 month, was it 2 months? That's part of the argument against continuous processing in biologics.
If you have a multi-product operation, then you have a failure of one of the unit operations within the room. It throws every other unit operation in the room into question, and you have to establish that there was no interaction between the problem. And that's going to be a pain in the neck to do. To solve the final stage of process validation, the one the FDA is really interested in my opinion, is going to be a real tough thing to do. And you're better off kind of crushing the problem. But single use has tremendous advantages for throughput, reliability, utilization, increasing scale out of processes and decreasing scale out. And that's the real advantage of single use technology. Not to mention the fact it's a lot easier. You don't have to establish completely closed single use. You can just use single use as single use.
All right, I think we're up against our 5 o'clock time frame here. If there's anyone that has
Everybody wants to leave.
burning question, we'll hang around here for a little bit, but officially we're at our time frame.
Yeah,
Thanks
everybody
very
else
much.
has planes
Thanks
to
very
catch.
much.
I don't have a plane to catch, and I'll answer any questions that anybody has.
Thank you.